Okay, so we're just gonna make a start at this time. Um, it's time for us to have a look of our week 7, which is the week when we're gonna have a look of the exam 2 and the lab practical 2. So as with the previous weeks, you will find the study guide for the exam 2 from your course learning modules for this week under the lecture study resources. Just because I'm in an area where there's a little bit unreliable signal at times, I'm just going to turn off the video, but that doesn't mean that I won't be available. I just will focus on sharing the screen at this time, and then as questions come up, we'll return the video on. So like I said, if you go to the study guide area and click there, you will be able to download or just keep on the screen the study guide that covers the chapters, the 3 chapters that we're looking for the exam 2. And as always, you'll notice that within the study guide, there's going to be empty gaps here and there. Whenever there's something that's empty, something that's missing in a study guide, that probably means that it's an important term, it's an important concept, something that I want you to take the effort of making a note of it. And as you remember, I will not be sharing the complete study guide at any other time than during the in-class sessions and these live online sessions. I will be, however, posting a recording of this live online session for you as well, in case you don't get a chance to join me in person. But as always, I'll also offer this session later tonight in the evening at 8 o'clock. The same content, the same things, but if questions come up, you can also join me for that. So having said all that, having shown you the study guide, what I'm going to do, just to be very efficient with the time, I'm just going to jump to the next window that I wanted to share. And the next window is going to be here. So we'll use this one to work our way through the important points, important things to notice from these chapters, as I said. And we started with the chapter on the heart. One thing to notice there is that whenever we're talking about anything to do with the heart, whether it's heart sciences, heart medicine department, or so on, you'll see people using the term cardiac to describe that. And let me just try to get my highlighter to work a little bit better. So the term cardiac is what you'll see being used when we're discussing about the structures that relate to heart, concepts that you might want to make a note of. Let's just try once more if we're able to emphasize. It looks like we're not. The other 2 things that I want to mention here, we started by talking about how heart was really human heart with its 4 chambers, almost like 2 hearts beating as one. So we have the left and right side. And for the left and right side, we remember that they directed the blood out of the heart to very different circuits. So if we look at the patient's right side of the heart, that's going to receive the blood from the systemic circulation. This blood is going to be deoxygenated, so low in oxygen, and it's going to pump it out to pulmonary circuit, so to the lungs. The purpose of this is that we're now getting the blood oxygenated again, and through this process of oxygenating the blood, we're now getting it back to the heart in a oxygen-rich form to the left-hand side. So that was our pulmonary circuit. Remember that even though in general, in the body, the arteries carried oxygenated blood and veins deoxygenated. This is reversed in the pulmonary circuit. So this would be the one time when that rule of thumb doesn't apply. Well, now we have the blood that has returned from the pulmonary circuit as rich in oxygen to the left-hand side of the heart. And there it passes through the structures that I've listed on the handout and leaves to the systemic circuit, so supplying all other parts of the body with the exception of the pulmonary circuit, so going to the lungs. This blood is now oxygenated, and once it returns from this systemic circuit to the heart, to our right-hand side, it's gonna be, once it returns back to the heart, it's gonna be now deoxygenated, so low in oxygen concentration. We also discussed a little bit about the location of the heart, and this would make a really good question in the exam. I think that there's a lot that we could ask about that. You remember we practiced some of that terminology that you're already familiar with. So rather than talking about chest cavity, are being located within the thoracic cavity, within the mediastinal place, between the space between the lungs. And we saw that in terms of directional terms, heart was superior to the diagram between the ribs 2 and 5, slightly left of the midline, so slightly left from that mid-sagittal line that you saw earlier on. I could also very well ask you about the apex of the heart. You remember that was the sharp tip, and that was pointing anteriorly, inferiorly, and slightly to the left. So that helped us to position the heart. And I'm just gonna share my video again. You remember we used the fist size as an approximation of the size of the heart. And when you pointed out the pinky finger, that was the line that pointed out from the apex. So anteriorly, inferiorly, and slightly left within your thoracic cavity. So I hope that that helps you to remember that. We also had a look at a few different kinds of layers, layer structures that we end up seeing. And we're going to start now actually by looking at the heart's muscle and moving from the innermost structures through the muscle wall to the more external outermost structures. You remember we had endocardium, myocardium, and epicardium. And it was really that myocardium that was the thickest meaty layer. I shared some examples of the research that really focuses on that. Another thing that we saw was that the heart was located inside a pericardium. This was the sac-like structure surrounding the heart and having the opening kind of tying up where we had the great vessels up here. So the purpose of this sac was twofold. First of all, it made sure that the heart had a chance to move about, as it is located there kind of snugly between the lungs. So we're making sure that there is room for the heart to do its movements. The other thing is that we're wanting to separate these different compartments to make sure that if there is an inflammation, if there's any other issues, it doesn't spread uncontrollably from one part of the body to another. So we're seeing that again, protecting the heart. So looking at the layers of the pericardium, again, working our way from the most inner one to the most outer one, we're going to see visceral and parietal layers. Between these two, there's going to be the pericardial cavity. You remember there was a small amount of fluids, and the purpose of this fluid was to facilitate the smooth sliding of these layers past each other, so lubricate that. I think we've discussed the next section already in terms of the movement, separation of the structures, and kind of making sure that any potential inflammation can't spread. One thing that I do want to highlight here is the inflammation of pericardium. So you remember in anatomical terminology, always the inflammation was itis, and in this case, it's going to be pericarditis. Well, a couple of things to notice about that. We could hear it with a stethoscope quite often as this crackling sound. As a result of this condition, we do end up with excess fluid leaking into this pericardial space and fluid building up there. And of course, in a long-term effect, we're going to end up with the heart's pumping ability getting compromised. So those would be some of the good things to know about the pericarditis. Then we moved on and had a look of the cardiac muscle tissue and cardiac muscle cells. Typically, I would draw this out for you, and I think we're going to try it even though my screen isn't responsive. We'll just do a diagram together. I'll share my camera in just a second. But before of that, I do want to mention we've discussed our skeletal muscles, muscle tissue, skeletal muscle cells, as they related to the large muscles that generated movement in our body. We have also looked at smooth muscle when we discussed about the blood vessels, and we'll come back to that when we discuss about the digestive tract. So this time, our focus will be especially on the cardiac muscle tissue. And cardiac muscle tissue is, of course, found only within the heart, nowhere else. You remember that that was not under voluntary control, so involuntary. You don't need to stop and think about that, hey, I'm going to have a heartbeat now. It happens automatically. Well, let's have a look on a cellular level, what a typical cardiac muscle cell would look like. So a couple of things that you'll end up seeing. First of all, you remember that these cells were typically branched in their appearance. And within these branches that we saw, another thing that we saw was that there were intercalated discs at the end of the branches. So why do we have branches? Why do we have intercalated discs? Well, that comes down to what are we trying to accomplish with the heart muscle and the cells that make up the heart muscle. So we want to make sure that they all contract in uniform. So we want to have as much possibilities for these cells to pass messages from one cell to another. So for that reason, we have this structure where the branches join to each other and these intercalated discs facilitated easy exchange of materials, easy exchange of information from one cell to another. A couple of other things that you might remember when it comes to the cardiac muscle cell. We had a central nucleus, one of those, and we had the striations that were characteristic as well for this type of muscle cells. So I don't think that any of those are new information or something that you wouldn't be familiar from the past, but definitely a good way to get us oriented as we're talking about this portion of the material. So now that we are familiar with that, and we're familiar with the concept that this all aims for the mass of the tissue to contract in uniform to do this pumping of the blood from one chamber to another. Let's have a look of the flow of the blood through the heart, and we'll see some of the important structures here. So we discussed how the blood arrived, if we kind of went this in a process manner, starting from one end, ending to another end, blood collected to the heart, to the right atrium through superior and inferior vena cava, superior one draining the head, neck, upper limbs, and inferior vena cava, the remaining parts. Blood builds up to the right atrium, then travels through the right AV valve, which is also known as a tricuspid valve, to the right ventricle. Well, now that the blood has moved to the right ventricle, contraction starts and squeezes the blood out, but it cannot return back to the right atrium. So instead, it has to go to the pulmonary trunk. And it goes to the pulmonary trunk from the right ventricle through the pulmonary valve. This was one of our two semilunar valves. From the pulmonary trunk, blood travels now to the lungs. Remember, on this right-hand side, of the heart, the blood is going to be deoxygenated. So the purpose why we're receiving it from the body is that we can get it reoxygenated at the lungs as part of this pulmonary circuit. Well, now that the blood has gone through the lungs, it's going to be reoxygenated, rich in oxygen, returning back to the heart, to the left atrium. So left side is going to be oxygen-rich blood. And from left atrium, blood travels to the left ventricle through left AV valve, or it might be also known as mitral valve, or it might be also known as bicuspid valve. So multiple options for the name there. And as you remember, I mentioned that students often wonder, do I need to know all of those? It would be beneficial. I could very well ask you in an exam to be able to provide alternative names for the same structure. So now that the blood has traveled to the left ventricle, this blood is rich in oxygen and we're eager to get it to the rest of the body where it can be utilized. So left ventricle, you remember we had a thicker wall there. As left ventricle contracts, blood cannot return anymore back to the left atria, so blood has to go to the aorta. And it goes there through the aortic valve. So second of our two semilunar valves that we saw. And now from the aorta, this oxygen-rich blood travels to all parts of the body other than the pulmonary circuit, does its job and comes back to the heart as deoxygenated blood back to the superior and inferior vena cava that we saw earlier on. So I think that that's a rather nice summary. We can see the same concept explained here in this sketch that I provided. And as you remember, I said that the simplest way of drawing a heart that I would totally be okay with, if you felt that that's the way you want to do it, is just to draw a box and divide it into 4 sections. So you see again, we have our right and we have our left side of the heart. And in addition, on each side, we have one chamber that's more superior and one chamber that's more inferior. And the superior chambers were always known as atrias and the more inferior ones as ventricles. So let's do the same journey that we saw just earlier on and start with the blood coming from superior and inferior vena cava to the right atrium, then traveling from there to the right ventricle through right AV valve or tricuspid valve. This refers to the number of leaflets or cusps, as you remember in this structure. Well, as the right ventricle starts contracting, blood cannot return back to the right atria. It has only one way to go, and it's going to be to the pulmonary trunk through the pulmonary valve. Notice that this line that I've drawn here is kind of a little misleading. Blood, of course, wouldn't go back to the right atria, so it would be more of a dotted line that you see there. Well, now that the blood that was deoxygenated to start with has been at the right side and has gone to the lungs, it's gotten now reoxygenated, so rich in oxygen, and it's going to return back to the heart to our left atria. So rich in oxygen after having been at the lungs. And from left atria, blood travels to the left ventricle through this valve that we see here, and we had 2 different names for it. Remember, left AV valve, or we could also talk about mitral valve, or we could also talk about bicuspid valve. So there's multiple names for the same structure there. From the left ventricle, blood, as it started contracting, blood again couldn't go back to the left atria, so it has only one way to go, and it's going to be to go to the aorta through aortic valve. Do notice again that the blood just travels from the left ventricle directly to the aorta, not through the left atrium anymore. So these 2 lines that you see going through the atria as the second lines going out of the heart, they probably could be dotted lines. So I might just do that update for our diagram there later on. Another structure that I do want to highlight, you remember interventricular septum separating the 2 ventricles. So between the 2 ventricles, the wall that we find there. And here I have a little bit more anatomically correct diagram of the heart with the same structures labeled and same concept. So let's just walk through it real fast together. So we had our inferior vena cava and superior vena cava bringing blood from the systemic circulation to right atria. This blood is now going to be low in oxygen concentration. This blood travels from the right atria to right ventricle through our right AV valve or our tricuspid valve. You'll notice as the right ventricle starts contracting, this valve that separated the atria and ventricle would slam close, and only direction where the blood could go would be to our pulmonary trunk through the pulmonary valve. So from the pulmonary trunk, this deoxygenated blood travels to the lungs, gets reoxygenated, and comes back to the heart through one of the 4 pulmonary veins. Pulmonary veins opened to left atria, and separating the left atria and left ventricle, we had our left AV valve, also known as mitral valve, and also known as bicuspid valve. So blood can only move to one direction from here. As the left ventricle starts to contract, the left AV valve has to slam shut. That's preventing the backflow of the blood. And only direction where the blood can go from the left ventricle is to the aorta through the aortic valve that we see here. Another thing to notice on this anatomically more accurate diagram is that pay attention to the left ventricle wall and how much thicker it is in comparison to the right side. Remember, on the right side, we didn't need as much force, we didn't need as high pressure because the pulmonary circulation really is not that far. Lungs are not that far from the heart, so we don't need as much force. Whereas with the left ventricle supplying all other parts of the body, we wanted to have that force for that blood to make its journey to even the smallest of the capillaries furthest away. 2 other structures that you see nicely on this diagram are the apex, so the sharp tip of the heart, and again our interventricular septum, the wall separating the 2 ventricles. So I hope that that makes sense. And now that we've talked a little bit about the valves, I think we should take just a moment to appreciate the purpose of them. So the purpose of the valves was to facilitate one directional flow of the blood, and the opening and closing of the valves was of course driven by these pressure changes within the chambers. 2 clinical conditions that I wanted to highlight here as they relate to what we've been looking at. If we have a valve that doesn't close properly, we end up with some of the blood being able to leak back into the direction where it shouldn't. So kind of the blood gets pumped back and forth, the same blood, without accomplishing this perfect unidirectionality. So that would be a problem with a failed valve. Another example that I've highlighted here was a narrowing of a valve when the valve does not open completely, and as a result of only partial opening, now heart has to work harder to pump that blood through that valve, and as a result of that, of course, we end up with insufficient flow, more work for the heart, and so couple of other structures. Some of these I have mentioned before in greater detail, some of them just in passing. You remember auricles were these projections from the atria, so both on the right and left-hand side, that can be used to increase the chamber volume if we fully inflate those. Trabecular carinae were just adaptations on the myocardial wall or on the heart muscle wall on the inside of the ventricles. These projections from the ventricular wall ensured that the blood couldn't pool there, stay behind, and start clotting. So we made sure that we got all that out. The term septum we've seen before, separating structures. We discussed about the heart valve failure in a class, and I proposed some solutions for dealing with those. And of course, one that we looked at was recovering heart valves from a donor of the same species, so from a cadaver after someone has passed. These allografts are used and quite well available if there's a greater need than what is available, but we're lucky there are areas where there's far less availability because of religious, because of other beliefs that restrict the use of the donor valves. The issue with this was, of course, the tissue rejection that we had to consider. Another option was to get these valves from an animal, in this case, with the xenografts. Pig heart valves are commonly used. We might need to do some further work there, for example, radiating these valves to try to make the fit as good as possible in terms of reducing the risk of rejection reaction. So that was one approach. The third approach that we discussed was to implant a mechanical valve to replace one of our natural ones. There were a couple of concerns that we had with that. First of all, anything that is man-made mechanical eventually will have a risk of breaking and failing. The other thing that we saw, if we have an individual, say, for example, a younger patient, and we replace a heart valve with a mechanical valve, and they keep growing and their heart keeps growing, this mechanical valve would not be able to respond to that in the terms of the growing with the rest of the heart. A few more other structures that I've mentioned here, two of these, the first and last one, especially relating to the fact that if you think during the embryonic development, there is not much point for the blood to be pumped to the pulmonary circulation since the lungs are not actively exchanging gases. So it's kind of a redundant work. And how we get around that is through these structures. So we have our fossa ovalis, which is what we can see still in an adult heart as a remnant of this embryonic bypass route for the blood to go from the right atria to left atria, so bypass the pulmonary circulation. So we can see this depression even later on in your life within the heart, this opening, which is full opening during your early part of the life when you're still developing before the birth, during the prenatal stages. That's closes completely the moment you are born and take your first breaths and so on. Ligamentum arteriosum was the other option, this kind of a embryonic remnant of bypassing tube from the pulmonary trunk to the aorta. So another one that we can sometimes see. We also mentioned coronary sinus. Coronary sinus was located within the right atria. That's where the blood from the coronary circulation comes back. So that was another depression that we can see there on the wall. So let's have a look at what else could we want to highlight. We're going to look at the surface anatomy of the heart. So remember, just like with the brain, we had sulcuses. These were the kind of depressions on the surface of the heart grooves. And there were three that I wanted to discuss in particular. And let's go through these together. So the first one, which I think we have discussed in your textbook with the name of atrioventricular groove or coronary sulcus, that was responsible for separating the atria and ventricle from each other. We also had on the anterior side a sulcus between the ventricles, so that's going to be our anterior interventricular sulcus, and similar on the posterior side, so posterior interventricular sulcus. The reason why these were important ones for us to learn was because of what lies in those. So let's have a look at those. On the left-hand side, or the left coronary artery that comes out doesn't actually run as a left coronary artery for very long. Very soon it divides into two branches. The left anterior descending, or as your textbook calls it, anterior interventricular artery, ran into this anterior interventricular sulcus. So it served the purpose of supplying most of the anterior portion of the heart all the way to the apex, the tip of the heart. The other branch from the left coronary artery was our left circumflex. Left circumflex ran within this coronary sulcus, wrapping around the heart, running to the posterior side. And finally, we have on the right-hand side, the right coronary artery, which ran on the right coronary sulcus or right atrioventricular groove, then going on the right side, having branches that lead from there to the anterior side and going all the way to the posterior side. So that's a good set of coronary arteries to see. I mentioned that we didn't spend too much time on the coronary veins, but the arteries are very important. If we look at the physiology of the heart, a couple of things to note there. Remember, it's going to be involuntary contractions. And if we compare this to the action potential of the nervous system cell, nerve cell will start seeing some interesting things. So just as a reminder, let's sketch quickly what a nerve cell action potential looked like. So we had this resting potential here that was about negative 70 millivolts. So this resting potential was established by the pumping of the materials within the nerve cell to generate slightly negative charge. And then we could end up with an event that was known as depolarization. During depolarization, the contents, the insides of the nerve cell became more positive, approaching the 0 millivolt threshold and probably overshooting it a little bit as well. So we became depolarized. After that, there was a brief period of plateau during which we have this charge close to 0 or even slightly positive until we are going to repolarize, go towards back to that negative 70 or even a little bit below that. And that little undershoot gave us that period of time during which another action potential couldn't progress. So what we knew as refractory period. The other important thing to notice about nerve cell action potential was, of course, that we're talking of a timeline of somewhere between 2 to 3, even to 5 milliseconds, but a very, very brief duration for these action potentials. So let's compare that to what we see with the membrane potential of the cardiac system. We are seeing the same events on this screen that I'm sharing, the depolarization, going towards 0 and a little bit above, and repolarization going back to the resting value. Big thing there to notice is that the resting potential, which is now known as our pacemaker potential, that's not running on a straight line. That's actually constantly climbing slightly from around negative 60 to negative 30 and negative 40. I apologize. And the negative 40 was the threshold where we saw the propagation of depolarization to be triggered. So we wanted to make sure that we started that heartbeat always. So that's why the pacemaker potential is constantly rising towards that threshold. We also discussed a little bit about the passage of the electrical impulses through the heart. And here we have a very nice diagram. So it was the SA node, a sinoatrial node that was our pacemaker located at the superior portion of the right atria, and from there the signals passed through our atria on both right and left hand side, and as the electrical messages passed through these structures, these atria contracted. As they contracted, they pushed the blood out from the atria to the ventricles. Now we have this group of cell bodies located between the right atria and ventricle in a location that we call as AV node. Within this AV node, we're generating a little bit of a delay for the electrical signal before it can propagate further. This delay serves the purpose that the atria have sufficient time to empty the blood out of them before we move further. So now, after this delay has passed, an AV node lets the signal propagate further. The signal travels down the bundle branches and Purkinje fibers, and eventually, once it reaches the apex, back up on the ventricular walls. And as the ventricular walls receive this electrical signal, the ventricles contract and start squeezing the blood out of the heart. So I hope that that makes sense. And a couple of things that can go wrong. We can have something known as arrhythmia, which was irregular heart rhythm, or fibrillation, which was really rapid, irregular contractions that would then cause for the circulation no longer being successful and this pumping of the blood through the heart in a controlled, one-directional manner being compromised. So how we treat that is through doing something that we know as defibrillation. So defibrillation is used to establish this strong electrical signal to the heart and heart's conduction system, and purpose of that is to establish the rhythm that we had before. So we're kind of trying to shock the heart back to proper functioning. This worked well with automated external defibrillator. So those are fairly widely available around many public spaces and so on. Individuals who regularly would need to have their heart defibrillated might also have internal pacemaker implanted, and this would include a little computer that would analyze, detect if there's an issue, and then deliver this shock. So rather than having those pads of AED that were implanted, we actually have a metal cord that wraps around the heart to deliver that shock. In terms of regulating the heart rate, that was from the brainstem, from the medulla, where the cardiac centers were located. Then messages traveled down the spinal cord and spinal nerves to the SA and AV nodes. And if we had to slow down the heart rate, we typically see vagus nerve very active for that. Well, if you remember, I showed you just a moment ago the nerve cell action potential. And in addition to the nerve cell action potential, I've now shown the cardiac muscle cell action potential on the screen. So very similar events. We have the resting potential, we have the depolarization, and we have the repolarization. There's actually two things to notice from this, though. Notice the period of plateau is not actually a straight line, so there's a constant slight repolarization going on in the cardiac muscle cell action potential. The other thing that's worthwhile to notice is the timeline. So rather than talking of a timeline between 2 to 5 milliseconds, in a cardiac muscle cell action potential is going to be somewhere around 250 to 300 milliseconds, so significantly longer. And if you wonder why that would be the case, well, that's so that we can facilitate for the blood to flow from one chamber to another, so we give it a little bit of more time for that to take place. We also conducted some ECG or EKG measurements in the class. So this technique known as electrocardiogram or electrocardiography is simply a way of collecting all these electrical signals that we just saw and mapping them visually for us on a trace. So on a trace, we typically would see 3 different elements. We would see our P wave, which is the first bump on this trace. During the P wave, the sinoatrial node and atria depolarize, followed by what we know as QRS complex. During QRS complex, you know that there's going to be ventricles depolarizing and atria repolarizing. And finally, wrapped up with a T wave. During T wave, the ventricles are now repolarizing. In addition to looking for these events on the EKG trace, looking whether they look normal, whether they're present. We can also look at the intervals between these different events. Some of the terminology that's important for us to remember: the period of systole was the period of contraction when the heart was contracted, and diastole when the heart was relaxed. We can talk about individual chambers, or we can look at the ventricles at large. Cardiac cycle referred to one complete heart cycle, so one complete heartbeat. Two heart sounds that you could hear: the lub-dub, lub-dub, lub-dub pattern. The first sound, the kind of a lub one, was when our AV valves closed on the right and left side. And the second dub kind of a sound was when our semilunar valves, so our aortic and pulmonary valves, closed. We also discussed about the cardiac output. I provided there explanation how by multiplying the heart rate with the stroke volume, you can calculate individuals' cardiac output. What I really wanted to highlight here was that if we compare the normal resting cardiac output, which was about 5.25 liters per minute, how high we can go if the need arises. So we can go up to 20-25 liters per minute. If you remember that there were between 5 to 6 liters of blood in a human body, that's really remarkable how fast we're now circulating the blood through the body. I normally would in a classroom discuss a little bit about congestive heart failure. I'm going to leave that for you to have a look yourself. And I also do typically take a little time to discuss about the embryology. Those who joined me in person got a fairly good comprehensive explanation of that. So I'll leave that for you to have a look as well from the recordings. In terms of the blood vessels, just in the interest of time, what I want to say there, we remember that we had 5 that we wanted to pay attention. We're going to move through them in the order as blood leaves the heart. So we have our arteries, arterioles, capillaries, venules, and veins. And remember, the capillaries were the main site of exchange of materials. Well, one thing that we saw, these blood vessels, regardless of what they were, they had a general structure that was shared by all of them. So tunica intima was the innermost layer in an intimate contact with the blood, if you wish, typically made of the endothelial cells of simple squamous type, so one layer of these flattened disc-shaped looking cells. The function really to facilitate twofold purpose: passage of the materials across this layer, but also protecting the layers underneath. The middle layer, known as tunica media, was the thickest of the layers. We're going to see a lot of smooth muscle and some elastin there as well. And the function of the tunica media was to control the diameter of the blood vessel, so doing vasoconstriction and vasodilation. The outermost layer was tunica externa, or also known as tunica adventitia. We find a lot of connective tissue at this area, also nerves, lymphatic vessels, and so on. And the function of tunica externa or tunica adventitia was to really reinforce these walls and anchor the blood vessel to its site. If we had an especially thick blood vessel, we could end up with a situation where the blood supply to the blood vessel wall itself was not sufficient through the from the lumen, and we needed to have blood vessels supplying blood vessels. This was the adaptation of vasovasorum that we discussed in the class. Couple of notes about arteries. Remember, arteries are immediately after heart, so must handle a very high pulsatile blood pressure. So they are typically very thick, very elastic walls. 2 types of arteries that I wanted to highlight. Elastic arteries are the first ones immediately after the heart, often known also as conducting arteries, so simply carrying the blood from one area to another. Thick wall, very focused the lumen of facilitating the flow, so low resistance there. The structure of elastic arteries, not surprisingly, contains a lot of elastin. We don't really see vasoconstriction happening on the elastic arteries. Instead, this expanding and recoiling helped us to kind of maintain the pressure reservoirs. Muscular arteries instead were known as distributing arteries. So this is the majority of the arteries that we see. We have lots of smooth muscle within these thick tunica media and less so elastic tissue. So these muscular distributing arteries really play a role in choosing where the blood goes to, so doing vasoconstriction, directing the blood where it's needed. And the final one that I have here are arterioles. Remember when we talked about capillaries, we often don't map out each and every single small capillary on that one area. So instead, if I had two capillaries, some from arterial side, so from arterioles, and some to the venous side, so venules, this area where we have this bunch of capillaries, I would refer it simply as a capillary network. And arterioles are very important because within the arterioles, we have precapillary sphincters that choose that how much of the blood actually goes to this capillary network, or do we just direct the blood through that area? So these little muscle bands control the openings of these little vessels. So I hope that that makes sense. You remember capillaries, we have a low blood pressure there, often just a tunica intima layer, and it was the site of exchanging materials. Three types of capillaries that I want to discuss. We have our continuous capillaries. So here the wall of the capillary is uninterrupted. We find these continuous capillaries, for example, at the skin, muscles, lungs, central nervous system, blood-brain barrier. Fenestrated capillaries, we have little windows. So the word fenestration referred to the French language for term for window. We would find fenestrated capillaries, for example, at the kidneys, intestines, sites where hormone secretions would take place. And finally, the sinusoidal capillaries. Here we have the big gaps on the capillary wall. So notice as big gaps are missing, so there's big sinuses, the flow of the blood is going to be even slower. So we find this at the sites where there's a lot of exchange happening, such as liver, bone marrow, spleen, adrenal gland, and so on. I think we already discussed about the precapillary sphincters and the capillary beds. So I'm just going to jump through that, and we'll have a look of the veins there. So we're going to have a lower blood pressure there than in arteries, but there's still some blood pressure higher than in the capillaries for sure. So actually majority of your blood vessels are veins, and at one time majority of your blood is within the veins. We discussed the concept, how do we get blood back to the heart from the most distal parts? So for example, from your big toe against the gravity, and there was a lot going on there. So there's going to be three things that I want to highlight when we discuss about that. Well, the first thing is that the veins, they were much thinner, their walls, than what we saw with our arteries. So thin-walled veins. And because the veins have thin walls, all of the skeletal muscles that are around them can really easily squeeze these veins. And as the veins get squashed, the blood inside the vein gets squeezed upwards and towards back to the heart. So this squeezing of the thin-walled veins by skeletal muscles was one thing that there's already two things, the thin wall and the skeletal muscles doing the squeezing that I wanted to highlight. Remember, we're constantly a little bit of moving, so this constant little movement helps to pump the blood, squeeze it on within the veins backwards towards the heart. Similarly, the big respiratory movements, especially in the thoracic cavity, contribute to this significantly. Other thing that we saw with the veins was our valves. So the function of the valves, similarly to the hearts, similarly to what we see at the heart, and let me just get my pen to work, was to prevent the blood from flowing backwards. So having these valves made sure that as we are squeezing the veins, blood is moving upwards against the gravity. It cannot return back to the direction where we don't want it to go. We saw an example of this when a soldier, for example, in a parade stands with knees locked still for a long time. We might not be getting enough blood returning back to the heart. As a result of that, not enough oxygenated blood is leaving the heart to the brain, and brain gets concerned. And the best solution to that is let's flatten this person. So now the brain and the heart are at the same level, and we can get a lot of blood flowing into there. So individual passes out. Lifting the feet of an individual who has passed out also helps us to make sure that maximum amount of the blood now flows to the heart and to the brain. So that is going to be your extra credit question in the exam, to be able to discuss about venous return, all those things that I mentioned also as it relate to passing out and so on. Another term that we see here, anastomosis, refer to collateral circulation. So these interconnections from one blood vessel to another to find an alternative route if needed. We discussed a little bit about blood pressure. Blood pressure measured in two values. The higher systolic value is the value we get during the contraction of the heart, and lower diastolic value is the value we get during the period of relaxation of the heart. Typically 120 over 80 millimeters of mercury. Few factors play a role: the volume of the blood, cardiac output, the resistance in the vasculature, and elasticity of the vessels. Remember, we often get a little bit higher blood pressure reading when you go to see a clinician or a doctor due to that nerves of the being present there. Pulse was measured, roundabout way of measuring the heart rate from the arteries based on this fluctuation of the blood pressure. And I've listed here multiple places where you could measure the pulse from, so pulse points for you to be familiar. We talked about the short-term regulation of the blood pressure using the homeostatic mechanism as an explanation tool for there. So we compared the blood pressure against the set point within the medulla. That was through baroreceptor, chemoreceptor reflexes, so carotid sinuses. So for example, we saw that at the where the common carotid divided into internal and external ones. We had receptors at the aortic arch, at the large vessels as well. And if necessary, we can then act on to stop that blood pressure fluctuation. Long-term regulation, remember that was through the kidneys, so renal responses by changing the blood volume, which would then change the blood pressure. Indirect mechanism through renin, angiotensin, aldosterone mechanism. So basically, we want to have an adequate tissue perfusion without damaging the vessels. That was the big goal. High blood pressure, hypertension, clinical criteria 140 over 90 had multiple negative outcomes associated with that, often multifactorial, whereas low blood pressure, hypotension, depending on what's causing it, cannot be less concerning, whether it's temporary, just getting dizzy when standing up, chronically low blood pressure, that's often associated with poor nutrition and it gives a risk for other conditions, or acutely low blood pressure, which could be a sign that we're looking of someone going to the shock. Signs and symptoms of the shock: pale, cold, and clammy skin, fast, shallow breathing, fast and weak pulse, appearing irritated and thirsty. And we talked about first aid treatment of the shock a little bit as well. And finally, the special circulations, I'll leave those for you to refer back to the lecture videos. I provided you a bunch of diagrams showing different kinds of circulations. These might be helpful for you as you are tackling the practical part of this week's exam. So I'll just leave them for you to have a look of independently. Finally, we discussed a little bit about the speed of the blood at different parts. Notice fastest at the aorta and arteries, lowest at the capillaries, veins somewhere in between. And this was a result of the total vascular bed area at these different areas. I'll leave the hydrostatic and osmotic pressure for our discussion of the lymphatic system, just noting that if this is not in balance, the hydrostatic and osmotic pressure, we end up with a buildup of fluid as an edema. So arteries are typically deeper, veins can be deep or superficial, and the anastomosis were those alternative routes. I'm going to stop it here at this time, and we will continue the last chapter the lymphatic and immune system chapter on our evening session. Until then, I appreciate you joining me, and I hope you have a very good rest of the day. And like I said, I'll make this recording available for you as well on the learning management system. Remember, I'm here to help you every step of the way, so just reach out if there's anything. Thank you. Bye.